Good afternoon, friends. I'm Saul. Today I'm with a new video of biochemistry course, Unit 3, Chemistry of Carbohydrate. By the way, this video is the final video of Chemistry of Carbohydrate. It contains glycogenesis, which is the formation of glycogen from glucose, and glycogenolysis, which is the breaking down of glycogen into glucose, and gluconeogenesis, the formation of glucose from non-carbohydrate molecules, hexose monophosphate shunt pathway, as well as regulation of blood glucose level are discussed in detail in this video. So please like and share this video and subscribe my channel. Let us go to the presentation. This is about glycogenesis. This glycogenesis is a process by which this glycogen is formed from glucose. Glycogenesis occurs in all tissues of the body, but majority site or the major sites of this process are liver and muscles. In the kidney also considerable amount of glycogen is produced from glucose through this glycogenesis process. Glycogenesis is a very essential process since excess glucose is converted and stored for later use. In the absence of this process, the tissue exposed to excess glucose immediately after meal, but they also starve it at another time. Therefore, this process can convert excess glucose into glycogen and stored in different body parts for later use. The following are some steps of glycogenesis. In step 1, glucose is phosphorylated into glucose 6-phosphate by hexokinase in the muscle and glucokinase in the liver. So this process requires the presence of ATP because phosphate group is obtained from ATP molecule. Here glucose is here and there is ATP, then one phosphate molecule from ATP is taken by this glucose molecule and it is attached on its carbon number 6. Therefore, in step 1 of glycogenesis, glucose 6-phosphate is formed from glucose and this ATP molecule. In step 2 of glycogenesis, this glucose 6-phosphate is then reversibly converted into glucose 1-phosphate. That means phosphate is shifted from carbon number 6 to carbon number 1, and this reaction is catalyzed by phosphoglucomutase, and this enzyme needs magnesium ion as a cofactor. In step 3 of glycogenesis, glucose 1-phosphate is converted into uridine diphosphate glucose. This glucose 1-phosphate contains phosphate molecule on carbon number 1, and this molecule reacts with uridine triphosphate. Uridine triphosphate is split into pyrophosphate and the uridine monophosphate. Then uridine monophosphate is attached to this glucose 1-phosphate and uridine diphosphate glucose is formed. Here, uridine diphosphate, 2 phosphate molecules are attached. One phosphate comes with uridine and the other phosphate is already attached to the glucose at carbon number 1. Therefore, uridine diphosphate glucose is produced. This reaction is catalyzed or activated by uridine diphosphate glucose pyrophosphorylase, an enzyme. In step 4, uridine diphosphate glucose is the immediate donor of glucose residues in the reaction, and this reaction is catalyzed by glycogen synthase, when the number of glucose residues more than 8, then glycogen is formed. Then this glycogen is either stored in the liver or in the muscle. Glycogenolysis is another metabolism of carbohydrate. When the blood sugar level falls, the condition is known as hypoglycemia. Glycogen stored in the liver and the muscle is broken down into glucose. By the way, this glycogenolysis is the opposite of glycogenesis. In glycogenesis, glycogen is formed from glucose, whereas in glycogenolysis, glycogen is broken down into glucose. To see some steps of this glycogenolysis, in step 1, this glycogen starts to lose its glucose molecules one by one. So when the first glucose molecule detached from the other glucose molecules of glycogen and an organic phosphate is added, therefore glucose 1 phosphate is produced, and this glycogen, which loses one of its glucose molecule, is remain. This reaction is catalyzed by glycogen phosphorylase and debranching enzymes. The combination of these two enzymes detached one of the glucose molecules from glycogen, and an organic phosphate is attached to the glucose molecule at the carbon number 1. In step 2 of glycogenolysis, this glucose 1-phosphate is reversibly converted into glucose 6-phosphate. That means this phosphate molecule is shifted from carbon number 1 to carbon number 6, and this reaction is catalyzed by phosphoglucomutase. In step 3, glucose 6-phosphate is converted into free glucose. This occurs in the liver and the muscle. The conversion of glucose 6-phosphate to free glucose is catalyzed by glucose 6-phosphatase enzyme. This glucose 6-phosphatase enzyme removes phosphate group from glucose 6-phosphate and enabling free glucose molecule to diffuse from the cell into extracellular fluid, extracellular space, including blood. Therefore, once glucose is free, it is diffused out of the cell and entered into extracellular space, including blood. This reaction, the conversion of glucose 6-phosphate to free glucose, is not occurred in the muscles because muscles lack enzyme glucose 6-phosphatase. Gluconeogenesis is another carbohydrate metabolism, and that is the process by which glucose molecules are produced from non-carbohydrate precursors or non-carbohydrate molecules such as lactate or lactic acid, glucogenic amino acids, glycerol, and propanol coenzyme A. The major site for gluconeogenesis is liver, and it usually occurs when the carbohydrate in the diet is insufficient to meet the demand in the body, or it may occur with intake of protein-rich diets, or it may occur at a time of starvation, or it may occur when tissue proteins are broken down and amino acids are produced. In these conditions, this process can occur in the body. Gluconeogenesis and glycolysis. This gluconeogenesis and glycolysis are opposing metabolic pathways that share a number of enzymes in common. In glycolysis, glucose is converted into pyruvate, and in gluconeogenesis, pyruvate is converted into glucose. It means glucose is converted to pyruvate in glycolysis, whereas pyruvate is converted into glucose in gluconeogenesis. Gluconeogenesis involves several enzymes of glycolysis. Of course, there are enzymes that are not used in common. So these enzymes that are not used in common are designed as key enzymes of gluconeogenesis. To show enzymes that are not commonly used by these two metabolic pathways, here glucose is converted to glucose 6-phosphate in glycolysis. This is the first step, and this reaction is catalyzed by glucokinase. But the conversion back of glucose 6-phosphate to free glucose is catalyzed by glucose 6-phosphatase. So at this point, these two metabolic pathways use different enzymes. Second, the conversion of this fructose 6-phosphate to fructose 1,6-bisphosphate is catalyzed by phosphofructokinase in glycolysis, whereas the conversion back of this fructose 1,6-bisphosphate to fructose 6-phosphate is catalyzed by fructose 1,6-bisphosphatase. Third, the conversion of phosphoenolpyruvate into pyruvate is catalyzed by pyruvate kinase in glycolysis, whereas the conversion back of this pyruvate to phosphoenolpyruvate is catalyzed by pyruvate carboxylase. Therefore, these points, these two metabolic pathways use different enzymes. Otherwise, the rest 7 steps are use the same enzymes for both metabolic pathways. Here we'll see one special type of pathways for which glucose is oxidized. It is hexose monophosphate shunt pathway, or it is also known as pentose phosphate shunt pathway. This hexose monophosphate shunt pathway or pentose phosphate shunt pathway is the same, or they are similar. Instead of some glucose molecules going through glycolytic pathway, it is shunted through this pathway. You know that glucose is passes through glycolysis pathway to produce pyruvate, but some glucose molecules are shunted in new pathway using a new pathway. This new pathway is known as hexomonophosphate shunt pathway or pentose phosphate shunt pathway. In the glycolysis, there is few bisphosphate intermediate product produced. For example, fructose 1,6-bisphosphate is a bisphosphate intermediate product in glycolysis, but in hexose monophosphate shunt pathway, only monophosphates are produced. Therefore, the name is known as hexomonophosphate pathway, or it is hexomonophosphate shunt pathway. Since the first carbon atom of a glucose molecule is liberated as carbon dioxide, so this pathway is known as the carboxylate pathway of glucose metabolism. As you know, in glycolysis, there is no formation of carbon dioxide. But in hexose monophosphate shunt pathway, as this carbon dioxide is liberated, so it is the carboxylate pathway of glucose metabolism. 10% of glucose molecules per day are entering through this hexose monophosphate shunt pathway. Especially liver and the red blood cells metabolize about 13% of glucose molecule by this pathway. The major purpose or the major function of this pathway is to generate reduced NADPH. This NADPH has the same function with that of NADH. They donate hydrogen and electron in reaction. NADH donates hydrogen and electron in catabolic reaction, whereas this NADPH donate electron and hydrogen in anabolic reaction. Otherwise, they have the same function. And in this pathway, pentose phosphate is produced. This molecule is important for the formation of nucleotides. As you know, nucleotides are monomeric units of nucleic acids. So indirectly, it is important for the formation of nucleic acids. Finally, about regulation of blood glucose. How our body regulates the normal blood glucose level. One of carbohydrate metabolism disorder is diabetes mellitus. Normal blood glucose level is known as euglycemia, which is between 16 to 114 milligram per deciliter. Too little glucose means hypoglycemia. In this condition, cells starve glucose, and that too many glucose is known as hypoglycemia. This creates sticky paralyzing effect. However, our body balance this blood glucose level in a number of ways. The balancing between hormones of pancreas, intestine, brain, and adrenals is required to maintain normal blood glucose level. For instance, regulation of blood glucose is largely done by the hormones of pancreas, which includes insulin, glucagon, somatostatin, as well as amylin. These enzymes used to regulate the blood glucose level. For example, this insulin converts excess glucose in the blood into glycogen, and this glycogen will be stored in the liver and muscle of the body. So this insulin lowers the blood glucose level, whereas glucagon helps to convert glycogen to glucose, and it helps to elevate the blood glucose level. Somatostatin acts as a policeman, balancing the production of this insulin and glucagon. For example, as food containing carbohydrate is eaten, it is obvious that blood glucose level rises. So pancreas turns on insulin production, and it turns off glucagon production. As a result, glucose from bloodstream enters into liver cells and converted into glycogen, and glycogen will be stored. But during starvation or during fasting time, when blood glucose level falls, this insulin production is stopped, or it is off, but glycogen production, glucagon production is on. As a result, this glycogen is hydrolyzed into glucose, and the glucose molecules are produced, so blood glucose level is right. Therefore, this somatostatin controls the production of insulin and the glucagon. That means indirectly it controls blood glucose level. The other hormone is amylin. This hormone is made in a ratio of 1 to 10 with insulin, and it helps to increase satiety, or it helps to increase satisfaction in the state of fullness after from meal, and it helps, or it prevents overeating. These are hormones that help to regulate our blood glucose level. Thank you for watching, and I will come back with another new video, mainly which focus on chemistry of lipids. Thank you, and goodbye.